One of the more frustrating things about viruses are the way that they are classified. Throughout the decades and centuries, we have classified viruses using a number of different systems, and it has led to a lot of confusion in how they're named. Viruses have been named, for example, by the disease they cause. You can think about, for example, the measles virus getting its name because it causes measles. Viruses have been named by the geographic area in which they were discovered. Think about the Ebola Zaire virus. Viruses have gotten named for the people who discovered them. The Epstein-Barr virus is an example of that. Viruses have been named by their morphology, by their shape, and the coronavirus is a good example of that. If you translate that term, it means crown virus. And indeed, under the microscope, the coronavirus looks like a sphere that is surrounded by a crown of spike proteins. Viruses have even been named according to the parts of the body that they infect. Neurotropic viruses, for example, like polio. Polioencephalomalacia is the disease that the polio virus causes. There is one system of classification, though, that we can apply to all viruses, and that system is referred to as the Baltimore System. The Baltimore System classifies viruses according to three things: according to their type of genome, how that genome is structured, a feature that we refer to as sense designation, and finally by their mode of replication. So the Baltimore System doesn't give a virus its name, but it allows us to compare one virus to another according to these three methods of classification: what kind of a genome it has, whether it's DNA or RNA, and whether that DNA is double-stranded or single-stranded, whether that RNA is double-stranded or single-stranded, what sense the genetic material is in, positive or negative, and what kind of system or process is used in the host cell to replicate the genetic material. Now, this is a very busy slide, but it basically lays out for us the different options within the Baltimore System. So if you start at the top, you can see genetic material laid out in all of the possible forms that we see inside a viral particle. So the most understandable to us as humans is double-stranded DNA. That's what they're showing us here. So there are viruses that contain double-stranded DNA, and this happens to be in what we call, again, the positive sense. Now, I don't want to get too far into the weeds of sense designation, but if you look on the slide down here in the middle area, they're showing us a messenger RNA molecule that was made from this DNA. Remember, this is a process that we refer to as transcription, where DNA is used to make an RNA. And you'll notice that this DNA with its plus sense matches with this RNA that has a plus sense. What they're saying is that the base sequence in this DNA actually is the same as the base sequence in this RNA. Now that's unusual. Normally we expect the sequence of bases in DNA to be complementary to the base sequence in RNA. So when we say that the DNA inside of a viral particle is in the plus sense, we're saying that the sequence of bases is identical to an mRNA that would be made from that DNA. That's unusual. if we had DNA in a viral particle that was in the negative sense or the minus sense, we would be saying that the sequence of bases in that DNA is complementary to an mRNA that would be made from it. So a minus sense or a negative sense designation actually makes more sense to us as students of cellular life, because that's what we see in a living cell. So we see all varieties of sense designation in viral particles, just as we see all varieties of types of genetic material. Double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA, and again, different sense designations. Remember, the other part of the Baltimore classification system is what process is being used in the host cell to replicate the genetic material, to make copies of the genetic material. If a host cell is infected with DNA from a viral particle, the host cell, remember, cannot tell its own DNA from the viral DNA. So the host cell would use the ordinary DNA replication process to replicate the viral DNA. That makes sense. But if a viral particle uses RNA as its genetic material, when that RNA gets injected into the host cell, the host cell can't take RNA and just copy it. That doesn't fit into our central dogma, does it? Remember, the dogma tells us that in a living cell, DNA can be used to make RNA, that's transcription. RNA is used to make protein, that's translation. And protein gives the cell its traits. We also know that DNA can be used to make more DNA through DNA replication. So if you're a virus and your genetic material is RNA, if you inject that RNA into a host cell, you're in a little bit of a quandary because there is no cellular process that takes RNA and uses it to make more RNA. It's interesting that the virus that is causing the current pandemic, and that virus's name is SARS-CoV-2. That's the name of the virus, and the disease it causes is COVID-19. The SARS-CoV-2 virus is an RNA virus. It uses a single strand of RNA as its genetic material. So when it injects that RNA into the host cell, our respiratory cells, our host cell can't just use DNA replication to copy that RNA. Something very unusual has to happen. And we'll talk about that in just a few minutes. So, when it comes to the Baltimore system, the key points to remember are the three things that it uses to classify viruses. And those three things are the type of genome, the sense designation of the genetic material, and the mode of replication. What process, what's the name of the process that the cell is using to replicate or make copies of the viral genetic material? All cellular life that's on this planet can be infected by viruses. That seems unusual to us. But yes, not only are eukaryotic organisms susceptible to viral infection, plants and animals, but prokaryotic life is susceptible as well. So the bacteria, the archaea, and the eukarya can be infected by different types of viruses. However, viruses are host specific. In other words, the tobacco mosaic virus cannot infect human cells. And that's because in order for a virus to infect a cell, the cell has to have a receptor on its membrane that that viral particle can interact with, can attach to, bind with, and then inject its genetic material into the cytoplasm. Now, some viruses are even more specific than that. Some viruses are not only host specific, but they are cell specific. In other words, inside the host organism, there is only one cell type that has the correct receptor for that virus to bind to. Influenza is a good example of that. The influenza virus is only capable of binding to certain cells in the respiratory system. There are other viruses, however, that are host specific, but they are not cell specific. The Epstein-Barr virus is a good example of that. In other words, inside the host, the human, there isn't one cell that Epstein-Barr can infect. There are several different types of cells that Epstein-Barr can infect. And that's because the receptor protein that the Epstein-Barr virus binds to is found on multiple cell types. Now, one of the most important things to know about viruses is that while they are host specific, and while there's a certain amount of cell specificity for some viruses, all of that specificity is subject to change. And that's because viral particles mutate. Just like we've discussed for living cells, the genetic material inside of a viral particle is subject to mutation. And let's recall what that word means. All a mutation is a change, a permanent change in the order of the bases. Now, the good news for you and I is that mutations inside of viruses do not commonly lead to changes in host specificity. Yes, viruses mutate. The influenza virus, for example, tends to mutate each and every year. And that's why we have to have new forms of influenza vaccine every flu season. But the host specificity for the influenza virus doesn't change very often, and that's a good thing. Recently, we have experienced this coronavirus pandemic because we did have a mutation event that changed host cell specificity. We currently believe that this coronavirus, the SARS-CoV-2 virus, originated in a bat. So the host was the bat. A mutation event then caused a change in host specificity so that this particular coronavirus is now able to infect human cells. That's an uncommon event, a host-to-host jump. And that's good for you and I. That means that these kinds of pandemics don't occur very often. Another good example of a host jump is in avian influenza. Another virus that we fear may become pandemic at some point is a form of influenza that currently has birds as the host organism. And again, that's avian influenza. In recent years, over in Asia, we have seen some mutations in the avian influenza virus that have allowed that virus to jump from a bird host to a human host. And this is very alarming to us for obvious reasons. However, right now at least, the transmission of that avian influenza from bird to human seems to stop there. In other words, what we haven't seen yet is transmission from human to human to human with that avian influenza. So right now, under certain conditions, the bird flu has been able to jump from bird to people, but it has not yet mutated to the point where there's sustained transmission from person to person to person. And that's a good thing. That's a good thing for us. I'd like to switch gears here and talk about viral life cycles. As we said, all viruses, in order to infect a host cell, have to attach to a receptor on that host cell. When the viral protein makes contact with the protein receptor on the host cell, those two proteins are going to bind together, and that's going to cause a shape change. That shape change is going to allow the genetic material from the viral particle to get injected into the host cell. Sometimes whole pieces of the viral particle get injected along with the genetic material. Once the genetic material is inside the host cell cannot tell the difference between viral genetic material and other genetic material. So there's no way for the host cell to tell which DNA is its own and which DNA might be from the virus, or which RNA is its own and which RNA might be from the virus. The host cell sees DNA and RNA and does what it does with those molecules. So the host cell is going to replicate the genetic material of the virus. It's going to transcribe and translate viral genetic material just like it does its own. And it's going to create viral protein as well as its own proteins. One of the most interesting things about the structure of viral proteins is that they tend to self-assemble. Remember, in a eukaryotic cell, protein folding is so complex that there are actual helper proteins called chaperone proteins that help make sure that each protein folds into its own unique three-dimensional shape. What viral proteins have evolved to be are simple proteins that are capable of self-assembling, self-folding into the correct shape so that they can perform the function of a viral protein. We should note that some of the more advanced viral particles actually contain one more thing other than just genetic material, and that's preformed enzymes that are going to allow for unusual processes to occur in the host cell. One of the most well-studied preformed enzyme that you will find inside a viral particle is an enzyme called reverse transcriptase. Like the name suggests, this is a viral enzyme that is capable of performing transcription in reverse. In other words, it's a viral enzyme that can use an RNA molecule to make DNA. And we'll talk a little bit more about that in just a minute. There is a class of viruses called retroviruses. And by definition, what a retrovirus is an RNA virus, a virus that uses RNA as its genetic material, that is capable of inserting a DNA copy of its genetic material into the host's chromosomal DNA. So this is a virus that, rather than just have its genetic material copied by the host cell, it's going to take its genetic material, it's going to create a DNA copy of it, and then it's going to insert that DNA into the host cell's DNA. It's going to place its DNA into the chromosome of the host cell. Now, in order to do this, it needs this very special viral enzyme called reverse transcriptase. This enzyme is going to produce a complementary DNA molecule from an RNA template. One of the best examples, again, the best studied example of a retrovirus is the human immunodeficiency virus, or HIV. And of course, this is the virus that causes AIDS. On this slide, you can see a drawing of the AIDS virus. Again, one of the best studied, most researched viruses. You can see that this virus has a structure that we've seen before. It's spherical. It has a nucleocapsid, and it also has an envelope around it, and it has spike proteins. In addition, it has preformed reverse transcriptase inside of it. So this genetic material in here, this is not DNA, this is RNA. And when this virus injects the RNA into the host cell, the human host cell, which happens to be a white blood cell called a T cell, the reverse transcriptase enzyme is going to take the RNA, the viral RNA, and make a DNA copy of it. That viral DNA will then be inserted into the DNA of that T cell. As you probably know, HIV is an insidious virus because it's capable of sitting inside that host cell for many, many years before it causes disease. It's possible for a person to carry the HI virus, the human immunodeficiency virus, for up to a decade and not know that they're infected. And unfortunately, they can transmit that virus to other people during that time.